So hi, everyone. I'd like to welcome you to today's CAMBAM/QLS talk, which will be given by Allison Shaw from the University of Minnesota. Allison has actually been a local in Montreal for the fall term when she was on a Fulbright scholarship on sabbatical. And she worked with a colleague at the Université de Montréal. She generally works on various aspects that all have to do with movement or migration, but with different sort of correlated topics with it, such as mutualism or eco-evolutionary feedback or structured landscapes in migration. And today she will talk about the connection of migration to parasites and animal infection, as well as she's also been very active in investigating how our perspectives and the diversity of the scientists shape the way we do science and the way we reach results. So I'm gonna let her share her screen, and you're welcome to ask questions both during the talk or put them in the chat or wait till the end of the talk.
Well, thanks Leah for the introduction, and thanks for the invitation to talk to you all today. So yeah, I had the chance last year to be in Montreal, which was a lot of fun, and get a chance to work with Leah and with my collaborator at University of Montreal. So as Leah said, I think about movement, and basically all organisms move at some point in their life, right? Whether they're an animal, a plant, whether they're fungi or microbes, whether they live in aquatic or terrestrial environments, movement is incredibly ubiquitous. But despite this sort of base commonality, there's huge variation once we take a closer look. So some organisms, just pull up the laser printer, some like plants might move a single time as a seed in their lives. Others, like these migrants here, are moving across the globe sort of continuously throughout their lives. And so in my lab, we're broadly interested in why it is that animals move and why movement matters. So this is kind of like a broad schematic of the questions that we like to ask. We're interested in like the causes of movement, whether it's sort of internal factors, intra or interspecific interactions, or abiotic drivers from the environment. We're interested how this shapes variation in movement. So when and where organisms move and how, what fraction of the population moves. And then we're interested in why movement matters for individuals themselves. So like, what are the costs and benefits to moving, how movement affects your population or community, how it affects like things like population spread, and even how that can scale up to affect ecosystems. So today I'm going to be focusing on like one small piece of what we do, and that's thinking about one of the causes of movement patterns and migration in particular. So if we think about why it is that organisms move, typically we can sort of think about two sets of factors. So maybe you move because you're trying to find something you like. So you're trying to move to find good food resources, or you're moving to find mating opportunities like these salmon here. And then conversely, maybe you're moving to get away from something you don't like. So maybe this fish was attempting to avoid being eaten and failing. Maybe these salmon are moving to areas that have good mates, but are also trying to avoid competition. And then we also might move to avoid harsh climate, like maybe you're a fan of snow, maybe you migrate away in the winter, or maybe you move to avoid things like pathogens and parasites. So a lot of the work that I've done in the past like 6 or 7 years has really focused on this last factor, pathogens and parasites, as a driver of movement behavior. And that's not because I think they're necessarily more important than everything else on the slide. It's that I think they're massively understudied, in part because they're really hard to see, right? It's really hard relatively to see parasites, except for these are little ticks on this puffin here, unfortunately. But most pathogens and parasites are pretty hard to see, much harder to see than like snow or a bear trying to eat you. And so I think because that they're hard to sort of see the effect of, people tend not to study them. So broadly, we're interested then in how parasites and pathogens might drive movement behavior. And like many things in biology, the answer is it's complicated, or there's lots of different reasons. And so I want to start with like a framework that we had developed in collaboration with two of my Canadian collaborators. This is Sandra Binning, who's the collaborator I mentioned at University of Montreal, and then Dom Roche, who's at Carleton University. And this is a sort of a conceptual framework that we wrote back in 2017. And so the first idea is that maybe you might change your behavior as an individual before you even get infected. So this is this sort of prophylactic behavior. So maybe you avoid areas that you know inherently have more infection risk in them. You avoid eating in patches that you know might sort of put you more at risk, and you avoid individuals in your population that seem to be acting sick. So you might change your behavior before you even actually directly experience a pathogen or parasite yourself. The second idea is that maybe you modify your behavior once you're infected. So after you're infected, you might adjust your behavior, and examples of this include behavioral fever. So some lizards like this one here, once they're infected, they'll actually move to habitats that have warmer areas. So they'll actually go and sun on rocks, because exposure to the sun increases their body temperature, and higher body temperature actually helps them clear the infection faster. So they're actually behaviorally inducing a fever in their body to help them cope with an infection. And then two other ideas are migratory escape, so migrating to escape parasites, or recovery, migrating to recover. And these are the two that I'm really going to focus on a lot for this talk. So I'll come back to those on the next slide. And then the third idea is that, okay, maybe your behavior changes once you're infected, but it's not because of something that you, the host, want. It's because your parasite is manipulating you to do something. And so there's this really cool example here you've probably seen of these fungi that infect ants and make them sort of crawl up into the canopy, and then the ants die on a leaf, and then the fungal spores can sort of disperse in a way that's good for the fungus. So there's lots of ways that pathogens can manipulate their hosts and move them to either habitat that's good for the parasite, or make them come into contact with other possible hosts for them, or increase the probability that they're going to be eaten by some other organism to sort of increase transmission. So there's, in short, like I started with people don't tend to think about parasites as drivers of movement. And now I've shown you there's this conceptual framework where there's tons of different ways that we can imagine that parasites and pathogens might be really important in changing animal movement behavior. And so I think this is an area that is really ripe for modeling and theoretical approaches. So I want to zoom in on these two ideas, this idea of migratory escape and recovery, and explain them a little bit further before I jump into the modeling work that we've done. So just to be clear, I mean migration in the sense of like animal population migration, a round trip, predictable, seasonal movement behavior, not just migration in the population genetic sense, which tends to mean just sort of directed gene flow. I mean sort of seasonal movements. And so that can be something like if you think about caribou that migrate, or birds that migrate, that sort of thing. So this first idea I wanted to introduce is this concept of migratory escape, where by migrating, you might actually escape infection from parasites or pathogens. So there's this paper in the 90s that proposed that one of the reasons that caribou migrate is related to their parasites. So caribou give birth in these specialized areas, these calving grounds, and some caribou, after they've given birth, they'll actually leave the area seasonally, and some stay there. And the challenge, like the area is really good for giving birth, but they have a whole bunch of warble flies. And so the warble flies will come and actually bite and infect the caribou while they're there. And so they've shown that caribou that migrate away from the calving grounds when they're not breeding have lower infection with these warble flies than those that stay. And so this idea was this term migratory escape was actually not coined until about 15 years later by this paper by Bradley and Altizer talking about it in a different context for monarchs. And the second idea that I want to introduce is this idea of migratory recovery. And so that is the idea that if you migrate along an environmental gradient, so you're changing the temperature of your environment or the salinity or humidity, that could actually promote recovery from the infection that you already have. So not only are you escaping pathogens or parasites, but maybe you actually are losing the ones that you have at a faster rate. And so there's a lot of work done on this, like this paper from the 1970s is describing flounders. So flounders migrate between freshwater and saltwater. they pick up parasites in saltwater, and the parasites will die more often and faster at lower salinities. And so one idea is that actually by moving across the salinity gradient, flounders are actually promoting their parasites to die. And so they're actually recovering from infection faster than they would be if they just stayed in saltwater. And so this term migratory recovery is one that I coined here with my collaborator, Sandra. Sorry, that should have a G at it. This is Sean Binning in our 2016 paper that I'm going to be focusing on for this talk today. So here's kind of an outline of where I'll go. We've done, I'll introduce, oops, there's a question in the chat. Oh, no question. I'll give you a sort of zoomed in exploration of this first paper that we developed, this model of migratory recovery in 2016. And I'm presenting it in detail because we've used that same framework to answer a ton of other questions. So the first part, this will really be the bulk of the talk, is I'll tell you, I'll go through all the details of the math that we did and the results that we found. And the second part, I'll give you examples of how we've been able to expand this modeling framework to ask a bunch of different questions, all about infection and migration. And then the third part, I'll zoom out a bit and tell you about how our perspective and what we view as important really shapes the science that we do. So this is where we're going. So here's a schematic of the model of, oh, I should actually give you our question. So we had this idea on the previous slide of migratory recovery, right? We saw this flounder example and we thought, okay, flounders are migrating between fresh and saltwater. They're able to lose their parasites faster when they move across that salinity gradient. But we know there's lots of other benefits to migrating. You know, animals are moving to find food or to escape climate and other things. We wanted to know, could recovery from infection ever be a strong enough driver by itself to favor the evolution of migration? And so this is a really hard question to answer empirically, right? Because any organism in the world is probably moving for multiple reasons. And so it's really hard to control for every other factor and determine empirically, does this organism migrate because of infection? But this is the exact kind of question that we can answer in a model. We can basically develop a model and say, the only reason why you might migrate is to recover from infection. Under what conditions is it beneficial to migrate compared to stay resident? So that's what we did. And here's a schematic of the model that we built. I'll show you a picture and then I'll walk through equations at the same time. So we imagine that the world has two environments in it. It has this orange one up top and this purple one down in the bottom. And then these white arrows are showing you the annual organism's life cycle throughout the year. So you can either stay in this orange environment for 2 halves of the year, I'm calling them T1 and T2, or you can migrate to the purple environment and come back. So we assume that anytime you're in the orange environment, you're becoming infected by a parasite or a pathogen. So we model this with a simple SI model. We assume that there's transmission from the environment. So we're tracking our susceptible and infected individuals, and we assume that there's just this constant rate of infection beta from the environment that's moving you from the susceptible class to the infected class. And again, so this is like you have some free living parasite that you're picking up rather than coming into contact with an infected individual. And so we assume that during the first half of the year, all individuals are in this orange environment, and we can just integrate these equations and write down the number of susceptible and infected individuals at the end of the first time period. So we just have our starting susceptibles, and then we're accounting for the proportion that did not become infected to tell us how many that are susceptible at the end of that time period. And then the number of infecteds that we have is we have the starting number of individuals that were infected, and then we add on all the susceptibles that became infected during the first time period. So next, we assume that there's this decision point here. So we assume that some fraction of the population, theta, is migrating to the purple environment, and the rest, 1 minus theta, are staying in the orange environment. And then we sort of write down the dynamics for each of those populations separately. So take the residents to begin with. So these guys that stay, 1 minus theta, we assume that during the second half of the year, because they're still in that orange environment, they're still becoming infected. And so their dynamics are still described by these equations, and we can integrate those and get to these two equations. So this is susceptible residents is a subscript and infected residents. Our susceptible residents is the number of susceptible individuals we started with at the end of T1 that did not become infected during the second half. And then we're going to account for survival. So our sigma is going to be the annual survival of susceptible residents. And then we're going to assume that infection has a survival cost. So this is, and then this 1 minus theta is accounting for the individuals that chose not to migrate here. And so same for infecteds, we're taking just the individuals that didn't migrate. We have our initial number of infected individuals, our susceptibles from T1 that then became infected during T2. And then we account for survival here, where we assume we take our baseline survival sigma, and then we assume that CI is the cost of infection. So infected individuals are more likely to die than susceptible ones. Okay, so these are our two equations for the residents that stayed here. Next, we can account for the migrants that moved to the second environment. And we assume that in the second environment, you're recovering from infection. So this could be like the flounder that's migrating from saltwater to freshwater, or it could be an organism that's migrating up a temperature gradient or something like that. There's no death. Oh, sorry, I don't, I'm a little behind on the questions. The death term, I'm not sure when you asked that, Anmar, but the sigma is accounting for the survival. I account for survival at the end of the year. If we did it.
You answered my question actually with the sigma and the CI, so it's okay.
Okay, perfect.
I asked the question before you introduced this. Sorry about that.
No, that's fine. I'm not on top of the chat. I'll try to keep it open this time. Yeah, we actually, I mean, that's kind of that's a technical question where like obviously organisms die during the year, but if you have population sizes change during the year, it makes it a lot harder to calculate things analytically, which we were trying to go for. So like realistically, you should imagine that there should be like death terms up here during the season, but we chose to account for it at the end. Okay, so we assume that you're in this purple environment, you're recovering from infection for this time period T2, and then you migrate back. And so we can again write down equations for this is susceptible migrants and infected migrants. So for susceptibles, we take theta, the ones that chose to migrate, the starting number of susceptible individuals at T1, and then we add the infected individuals who migrated and then recovered in the second environment. And then we account for their survival and mortality here. So we assume that infection can kill you. We also assume that migration can kill you. So migrants, even if they're healthy, are paying a survival cost from having migrated. And then for infected individuals, we have, again, the ones that migrated. We have the infected individuals that did not recover. And then we account for their survival here. And they're really unlucky because infected migrants have to pay the cost of migration and they have to pay the cost of infection as well. Okay, so we're kind of at this point in the annual cycle. We've accounted for survival, and then we assume at the very end of the year, reproduction occurs. So we assume that susceptible individuals have a potential maximum fecundity of phi, and infected individuals might potentially pay a cost of infection that reduces their fecundity phi. And then we account for density dependence with this kind of generic density dependent term. So B max is the maximum number of offspring that are produced. So each susceptible individual, resident or migrant, produces phi offspring. Each infected individual produces phi times 1 minus CF, the fecundity cost of infection. And then to get the realized number of offspring produced, we multiply this B max by a density dependence term. And we can actually do the whole model analytically without specifying the exact form of density dependence. We just assume that it behaves nicely like you would expect density dependence to, so that if the population size is 0, this term just goes to 1 and it kind of goes away. And then as you increase the population size, the density term decreases. And so as you have more individuals in the population, you have lower and lower fecundity until you're sort of at the highest maximum amount, and then your fecundity goes to 0. So if we come and we assume that all offspring are born susceptible. So if we bring together all these equations, we can actually write down a 2x2 matrix equation. So at the top here, I'm recording the susceptible and infected individuals, and now I'm indexing from 1 year to the next. So I showed you the dynamics within a year, and now we want to know from 1 year tau over here to the next year tau plus 1. We just multiply it by this 2x2 matrix with all these coefficients that are kind of given down here. And I've grouped the terms in this matrix by the theta, by the probability that you migrate, because that's the kind of the parameter that I'm most interested in focusing on. So if you recall, our question is really, under what conditions should an organism migrate in this model? And so really we want to know, what should the value of theta be? Should theta be 0, meaning that you don't migrate? Or should theta be 1, meaning that you migrate all the time? And then how does this value of theta depend on all these model parameters? How does it depend on your beta and gamma, your rates of infection, your sigmas, which here are your rates of survival, and then your fecundity and potential fecundity cost of infection. Okay. So to analyze it, the first thing we do is we look for the equilibria of the model, do the sort of ecological equilibrium. So like, what population size do we expect to see in the long term? And we're just looking for places where the number of susceptible individuals from one year to the next is constant and infected is constant. Like, obviously they're going to change throughout the year within the dynamics, but we're looking for constant from year to year. And so there's a non-trivial equilibrium, which is given by this. Again, it's kind of weird because we can solve this without actually specifying a density dependent term. And we can say that when the density dependence is exactly equal to this expression, and then we can calculate infection, number of infected individuals is just our susceptibles times this constant. This is not supposed to necessarily be intuitive. It's not intuitive to me, but just to show you that we can actually do all of this analysis analytically without yet resorting to simulations. So we have a closed form solution for like how big our population size is expected to be. And then the next thing we want to ask is really like, what theta value do we expect to see? What should the migration probability be? And we do this by looking for the evolutionarily stable strategy. And so that means the migration probability that if it was adopted by a whole population of individuals could not be invaded by a mutant playing a different migration probability. So I'm not sure how many of you are familiar with ESS concepts and sort of methods. So I'm going to give you a brief aside talking about ESS versus optimality. So a common approach, certainly in sort of biological models of math, is looking for like optimal traits. So if we didn't take an ESS approach, we could basically say, we have some trait we care about on the x-axis, theta, this migration probability. If we can describe fitness on the y-axis, the optimal trait will be the value of theta that gives you the highest fitness, right? So this would be an optimal approach. And this is basically considering an individual, this little red dot, in isolation. What an ESS does is sort of acknowledge that this little individual, a red dot, exists within a broader population context. These other blue individuals might be having different migratory behaviors, and they're going to affect this red individual's fitness. So the idea is that rather than fixing the trait, just looking at one value of trait, we have to consider, okay, we have a mutant that's red, that's playing some trait on the x-axis, and it exists within a resident population that's playing a specific value of this trait as well. And so this fitness landscape here, this gray line, depends on what the resident population is playing. And so here it would say that if this is your resident trait value, mutants that are playing a lower value of that trait will have lower fitness. Mutants that are playing a slightly higher value will have a higher fitness, and we assume that they'll be able to invade. And so we can say, okay, well, that's going to shift the resident trait slightly to the right. If we imagine it shifted further, it might look like this. So the underlying landscape is set by this, this up and down line, which is setting the resident strategy. And what we're looking for is a place here in the middle. And this is called evolutionarily stable. It's basically, if this is your resident trait, any mutant that's playing a different value is not going to do better than you are. Any mutant is going to have lower fitness than this resident and is not going to be able to invade. Or a way of saying that mathematically, if G is our growth rate or our fitness, the growth rate of a resident in its own population is going to be bigger than the growth rate of any other mutant in that resident population. Or if these two are equal, the growth rate of a resident in a mutant population has to be bigger than the mutant in its own population. Okay. So the way we do this mathematically is we write down a version of the model now where we're describing the growth rate of some mutant strategy, we'll call it theta prime, then we assume that exists in a resident population that's playing a different strategy, theta bar. So all this affects for this equation is that we assume that the density dependent term is set by the resident strategy. And then we calculate the growth rates just using the same model as before. And then we look for the place that's an ESS as defined on the previous slide. Sorry, I'm getting over a bit of a cold here. Okay, and we can actually solve this analytically as well. We can say that our evolutionarily stable migration strategy is this kind of mess of a thing. And again, this is not super intuitive to me, but just to show you that we can do it all closed form. So our theta could be 0, meaning that no one migrates. It could be 1, meaning that everyone migrates, or it could be some intermediate term here. We get solutions that are bimodal as well, which I don't think we actually see in this model. I'm not remembering exactly. And then these conditions and these XY terms are given by this combination of the model coefficients and parameters. So it's kind of an ugly mess, but it's just to show you that we can do the whole thing analytically. But I don't find it super intuitive to look at. I'm guessing you probably don't either. So I'll walk you through what the results actually mean biologically by looking at some pictures of the results graphically rather than looking at these equations. Okay, so first consider a really simple and really extreme case where infection happens really, really fast. So beta is really big in the first environment and recovery is really fast as well. So gamma is really big in the second environment. That means that these exponential terms basically go to 0, which makes our life really easy. And this theta ESS term just boils down to this. Either it's 0 or 1 based on the relative costs and benefits. Or if you want to see that picture graphically, if I plot the cost of migration on the x-axis and the cost of infection on the y-axis, what we see is that areas in black, if infection is really costly and migration isn't, you should migrate, right? Or down here, alternatively, in areas that are white, when migration is really costly, but staying and being infected is not costly, you shouldn't migrate. So this is pretty straightforward. Basically, whichever one is the less costly option, that's the one you should choose. Either stay and be infected or migrate and recover from infection. Now, obviously, this is a really extreme case, right? We're not always guaranteed to be infected, and we're not always guaranteed to recover in the other environment. So we can imagine what happens if we slow down those beta and gamma rates. What happens if we make them smaller? And we can see over on the right, when they're slower, this straight line now shifts to kind of curve up a little bit here. And just like kind of squinting at the left versus the right, you can see that on the right, there's a lot more white space. So not migrating is favored over a much broader range of parameter space. And that makes a lot of sense, right? We basically have said over here on the right, beta is really small. So you're less likely to get infected if you stay in the first environment. So you have less incentive to migrate. And gamma is really small, meaning that if you do migrate, you're a lot less likely to recover from infection. And so the costs of staying are less and the benefits of going are less. Overall, you should be less likely to migrate. So the first sort of key set of results is you choose whichever option is less costly, but also accounting for the risk. What's your risk of getting infected and what's your probability of actually recovering if you do migrate? Okay, so the second thing you probably noticed on the right is we now have this kind of rainbow region in the middle. So black is always migrate, white is never migrate. And then down here, these yellow to blue values are intermediate values of that theta parameter. So that's like, say you migrate 20% of the time or once in every 5 years. So to think about that, imagine a case where your infection risk is pretty low in a single year, right? So you stay in your environment, your orange environment 1 year, you're unlikely to become infected. So migrating isn't worth it. But say you live for 5 or even 10 years. Over that time, at some point, chances are you're going to become infected. And the way we've set up our model, the only way you can recover from infection is by migrating. We assume that there's no sort of baseline recovery at all. And so once you get infected, you have to pay that cost year after year unless you migrate. So therefore, it's probably beneficial at some point in your life, if you're long-lived, to migrate. But it's not necessarily beneficial to migrate every year And so we end up with this kind of mixed strategy where you hang out in the first environment for a couple of years. You probably pick up the parasite by then. And then you migrate once, recover, hopefully, and then you come back to your first environment, hang out there a couple more years, and so forth. And so what we can see is that if I plot beta on the x-axis, the rate of infection, and gamma on the y, you can see these mixed strategies are only favored if your infection risk is sufficiently low and your recovery rate is sufficiently high. So you have to have a low risk of infection and then a good guarantee of migration. So the question, does the cost of infection increase as a function of time if you choose not to migrate? No, but you, so let's see, your cost of infection is independent of how many years you've been infected. But if you're living for 3 years, you're having to pay 3 survival costs of infection. Does that answer your question, Rodrigo? Yeah. I mean, you could assume it, but then you'd have to like track how many years you've been infected, and the model would like, you could actually have it be like a more extended matrix model that was like, how many years have you been infected for? And you probably could actually do it. That would be kind of cool. Okay, so the results that I've been showing you so far have really focused on the survival cost of infection. But if you remember back when I introduced the model, I said you might also have a fecundity cost of infection. So now I'm showing you on the right side, infection only has a survival cost. And on the left, I'm showing you when infection has both survival and fecundity costs. And so again, if you kind of squint at this, you can see over on the left, there's a lot less white area, a little bit less white area than on the right. And that makes sense, right? On the left, I'm saying, if you're infected, it is both more likely to kill you and more likely to reduce your ability to produce offspring. So it has 2 costs. Whereas on the right, infection only has 1 cost. And so if infection has more costs, you should be more likely to avoid it and you should be more likely to migrate. So that at least that bit makes sense. But we also see something kind of funky. So as you go up the y-axis, you're increasing the infection cost of the parasite. And so over here on the right, as you increase infection cost, that's sort of increasing the selective pressure to migrate, and you're shifting from not migrating to migrating more. But over here on the left, there's a region here where as you increase the infection cost, you're actually shifting from a partially migratory species, you migrate every couple of years, to one where you never migrate. And that's weird, right? That's like basically saying, as you increase the cost of infection, you should be less likely to migrate, which is going to mean you're more likely to get infected, which means you're experiencing that cost. And that was like super counterintuitive. It took us a while to figure out what was going on. And it turns out that as you go up this y-axis, there's kind of 2 things that are changing at the same time. So yes, your parasite is becoming more costly. But second, as you increase the survival cost of infection, you're reducing the average lifespan of an individual. So if you're more likely to die from infection, you're going to live a shorter life. And I said a while back that like this partial migration strategy of migrate every couple of years, I think I said this, is only favored if you live for sufficiently long enough, right? So if your strategy is migrate once every 10 years, that doesn't really make sense unless you're living for like 10 years or so. And so as we move up this y-axis, we're reducing the average lifespan of individuals in the population, which means partial migration becomes less favored, and we actually see this weird counterintuitive switch from migrating part of the time to never migrating, even though infection is getting more costly. The question, the migration probability does not explicitly depend on infection, right? Yes. Yes, I will get back to that, Suresh. We actually did develop a model where it does depend on that, but that's a great question. We assume that your probability of migrating is the same for all individuals, whether or not you're susceptible and infected. But I'll come back to that in like 3 slides. Yeah, so Rodrigo, in other words, if I'm going to die soon, you might as well just stay. Yeah, exactly. I'll stay tuned. Keep that. Come back and ask me more questions in a minute. So usually when I give this talk to more biological audiences, they're like, great, you made a bunch of models, you showed this thing theoretically can happen, but like empirically, does this actually happen, or is this just something that you've made up? So there was a paper, so this was a 2016 modeling paper that we put out. And then in 2018, they actually did this really cool study where they put radio trackers on toads and they tracked their infection status throughout the year. Because really, to think about migratory recovery, you need to know an individual was infected, then they migrated, and then they recovered, right? Like that's kind of the first piece of information. And that means that you have to track not only infection status over time, but infection status over time while individuals are migrating. And so they did this with these spiny toads, and they basically showed that as they migrated between terrestrial and aquatic environments, they actually lost their BD infection. So moving to terrestrial environments is actually providing a refuge from infection. And they actually said in their paper very nicely, this is direct documentation of migratory recovery, which is pretty cool. And it's always exciting when you develop a model and like someone actually takes it and uses it to inform empirical work. Okay, so this is kind of the first part and the bulk of what I wanted to talk about today, to give you like a really detailed perspective of how we can think of parasites and migration as interacting in this really simplified modeling approach. And the second part, I wanted to talk about some extensions that we've been doing with this modeling framework in place to explore some other questions. So the first thing I did, this is a second paper with Sandra, who was on that first one, is if you were looking carefully, you'll notice that in this model that I presented, the orange environment and the purple environment, in the purple environment, you're recovering from infection, but you're actually also escaping infection, right? So actually, this gets back to your question a little bit, where like infected individuals benefit by migrating because they can recover from infection. But susceptible individuals actually also benefit by migrating because there's no parasites in the second environment. And so they're actually escaping infection. So our model, although we framed it as migratory recovery, had both escape and recovery in it. So what we did in this paper, this 2019 animal ecology one, is we built a model where we could actually turn on and off escape and recovery. And this was a so I've noted which things are simulated and which ones are analytic. And we basically ran a model and said, okay, if you only escape, how much migration should evolve? If you can only recover but not escape, how much migration should evolve? And typically we found that more migration evolves when you only recover but you don't escape than if you only escape but you don't recover. So it's interesting because if you look at the empirical work, people pay a lot more attention to this idea of migratory escape, and migratory recovery really has not been paid attention to. But yeah, at least on the theoretical side, it suggests that migratory recovery might actually be a stronger selective pressure on migration than escape. So the second thing we did, this is getting directly back to your question, is when should susceptible and infected individuals have different migration strategies? So this was work that was led by my grad student, Naveen Narayanan, and we basically wrote down the exact same model, but we said, okay, now there's two migration strategies, one that you play whenever you're healthy and one that you play whenever you're infected. And typically what we found is that it's much more common for you to stay in the environment when you're healthy and to migrate when you're infected. So this like mixed strategy that I talked about before is perhaps better described as a conditional strategy, not migrate every 5 years, but anytime you're healthy, you should stay, and anytime you're infected, you should migrate. So if you want to see more to answer those questions, this is an OICWIS paper from 2020. Okay, another thing that we wanted to ask is, well, what happens if infection intensity varies in the population? We built this really simple SI model, but it basically bins all infected individuals together. It says if you have 1 parasite or 20, it's basically the same. And that we know that that's not really true biologically. So what we wanted to do is basically develop a model where rather than having S and I, we had these infection intensity classes. So we had 0 intensity, which is basically susceptible, and then kind of like low, moderate, and high infection. And we said that if you're in the first environment, you kind of basically move up intensity classes as you accumulate parasites. And if you go to the second environment, you're not accumulating anymore. And this was work, I should say, that was led by a really awesome math undergrad I had, Lori Balstead, and was in collaboration with Marlene Zook and Megan Kraft, who are both faculty here at the University of Minnesota with me, and of course with Sandra, who's on a bunch of these. And what Lori found is that infection or migration should actually vary with infection intensity. And we predict that if you have a low intensity of infection, you should typically be migrating, whereas high intensity of infection individuals should stay. So this was a case where we assumed that you paid costs when you migrated and you paid costs when you were infected, but you had to pay a higher cost if you were more infected and trying to migrate. So there was kind of an escalating cost of infection, which is not quite the same as the earlier question about like longer terms of infection, but maybe it's similar, Rodrigo, to your question about how long you've been infected for. Let's see. Another thing that we've tried is usually I've gotten questions about like, okay, you've modeled assuming indirect transmission. Oh, sorry, going back. What did you say? So Rodrigo says, does the infection intensity depend on the time or the ratio of time in the infectious environment? It's the time, the cumulative time spent in the infectious environment. So as you're in the infectious environment, you're accumulating parasites according to like a saturating function. So the longer that time you spend in that first environment, the more you are accumulating, but you can get to a point where you are like so saturated with parasites that spending any more time there doesn't actually make a difference. Okay, so the other kind of direction we've explored is thinking about different forms of transmission. So we'd focused on this indirect one from the environment because the math was way easier. It was analytically tractable. But there's lots of parasites and pathogens that really you only get transmission if you have contact between susceptible and infected individuals. So there's density dependent, where as you increase population density, you're more likely to come into contact and you have increased transmission. There's also frequency dependent. So this is more of like vector-borne pathogens or sexually transmitted ones where S and I individuals have to come into contact, but the contact rate is more dependent on, or the transmission rate is more related to the frequency of infected individuals in your population. So unfortunately, if you switch to density dependent and frequency dependent, you get transcendental equations and you can't solve those, of course. So we've done this sort of comparison of these three different transmissions doing simulations. And one of the interesting things we found is that if we ignore recovery, if we just focus on escape, we see that this partial migration, that colored region where you only migrate part of the time, it only ever evolves if your transmission is density dependent and not under these other two scenarios. And that's because if your transmission is indirect, there's always a best strategy. It's either best for everyone to migrate or best for no one to migrate. But if transmission is density dependent, then if half of your population migrates, that actually lowers the transmission for individuals that stayed in the infected area. And so you can get these basically partial migration as kind of a way to split the population that lowers transmission for everyone. And then going back to frequency dependent, again, it's kind of like indirect, where there's always a best thing to do. Like if it's best for one individual to migrate, it's really best for everyone to migrate. And this was, again, work with Megan, Marlene, and Sandra, who I talked about in previous slides. Another thing we've gotten or sort of thought about is like, okay, well, I assumed that the orange environment has parasites in it and that magically in our model, the purple one doesn't have any parasites, right? And that's not realistic. We know that obviously all environments have parasites in them. So we built another model that said, okay, anytime you spend in the orange environment, you're infected with one kind of parasite. And anytime you spend in this purple environment, you're infected with a second parasite. Is it still actually beneficial to migrate or not? And this was work in large part done by another awesome math undergrad I had, Julie Sherman, with Marlene again, who I talked about, and with Keith Barker, who's also a faculty member here at University of Minnesota. And one of the cool things we found is that migration can still evolve if you are exposed to new parasites, as long as migration is lowering the prevalence of infection. So the probability overall that you get infected with something. And you end up with this cool contrast of results where migrants both have lower prevalence of infection, so a lower probability of getting infected or a lower proportion of the population that's infected. But at the same time, they have a higher diversity of parasites. So typically in this model, migrants will have both parasite 1 and parasite 2, whereas the residents that stay in this orange environment are only going to ever have parasite 1 because they're never exposed to the second parasite. So that's kind of like a very crash course in all the things we've thought about with this modeling framework. I'm happy to talk any more about them in detail, and I've added all the references there in case you want to go back and look at them. And so in just the last couple minutes, I want to talk about this third idea, kind of zooming way out. So at the start of my talk, I showed you this slide. I said, I am thinking about parasites because people basically haven't studied them because they're hard to see. And so therefore, because they're hard to see, people have typically assumed that parasites are really not important. So we generally don't take the perspective, for people like me who think about movement, do not take the perspective that parasites are important. And so if we never consider that parasites might be important for organisms, we're never going to ask questions about how parasites shape host migration, right? We have to start by considering that they might be important. And so the science that we do, the models that we develop, are really shaped by our perspective. When we build models, we put in what we think is important and we leave out things that we think are unimportant. And so if we always focus on things like competition or climate, then we're always going to be putting those things in our models, and we're always going to be leaving out other things. And we can't understand their importance if we don't put them in the model. The second thing, though, is that our perspective, the way that we view the world and what we think is important, is shaped by our experience, right? We were chatting about this at the beginning, but like when I first started working on this years ago, I had to really convince people that like parasites and pathogens might actually shape movement behavior of organisms that are migrating these really long distances. But then think about like what we've all lived through the past 2 years, right? These are just some headlines from the Delta wave of COVID. We've all dramatically changed our movement behaviors in response to this little tiny pathogen that we can't see. It's had a huge impact on everything that we do, and yet it's just a little tiny pathogen. And so the way that we view the world and what we think is important is really shaped by the experiences that we've been through, like COVID. I now suddenly have to, spend a lot less work convincing people that pathogens might be really important for behavior because we all have direct experience that they absolutely can be. But the second aspect of that is perspective is not only formed by our experience, it's actually formed by our identities, that who we are as individuals. And this is something that I think scientists rarely talk about, but I'll give you just 2 examples of people who have talked about it. So this is Karen Workington. She's a queer biologist who studies life history transitions in frogs, these little guys here. So she's interested in how they choose when to hatch, for example. So there's an interview with her, this 2017 one, where basically she says that the transitions in her own life, basically her coming out as queer, has really shaped the way that she views transitions in the organisms that she studies, right? Her own transition shapes the way she thinks about transitions in life. And here's a second example. This is Robin Wall-Kimmerer, who's an indigenous biologist and really an awesome writer. You should check out Braiding Sweetgrass. Is a great book if you haven't seen it before. So as she writes about in her book, her identity and her as a member of the citizen Potawatomi Nation has shaped her research interests in restoring human relationships with the land and humans as part of ecological communities. So these are 2 examples of people who have written about how their own identities as individuals is shaping their perspectives on the world and shaping the way that they do science. So I want to argue that perspective and identity also shape the way that we develop mathematical theory in biology. And this is something that I think we really don't talk about at all, because we like to think that math is objective and therefore math models are somehow objective. So let's see if I can convince you otherwise. So when I build a model, I view it as kind of like a 4-step process. I have some biological question that I want to ask. I can't get at the answer directly, so I have to translate it into a mathematical model. I analyze my mathematical model, and then I interpret the answer biologically. So in this kind of sense, developing theory, the work that I do, really requires 2 key steps of translation between the language of biology in blue and the language of math in red. So I know many of you are living here in Canada in a bilingual country, and I'm sure you all have experiences with how challenging translation can be, right? So, and any scholar that works on textual translation will tell you the same thing, that there's multiple different ways to translate the same text. So here, this is a great book that it's called 19 Ways of Looking at Wang Wei, and it gives 19 different translations of the same Chinese poem, and they're all different, right? So it's really clear from this book in particular that the perspective of the translator shapes the way that translations are done. It shapes what words they focus on, what aspects of the poem they choose to include, and which aspects they think are fine to ignore. So by extension, math modeling is effectively translation, right? And so thus it's subject to the perspective of the translators, me or you or whoever it is that's building the model. So another way of getting at this is thinking about two quotes. We also talked about this before the talk. I'm sure all of you have seen this one, the idea that all models are wrong, but some are useful. I think in the same way you could say that all translations are wrong, right? There's no such thing as an exact translation because you can't do that. No model is going to capture perfectly a biological system because it's in a different language. You just can't do that. But we don't talk about the corollary of this idea, or the quote that I like, which is, our truth is the intersection of independent lies. And so it's the idea that multiple translations of the same poem provides a much better understanding of the poem than any one alone. And similarly, multiple models of the same biological system provide much better understanding of that system than any one model can alone. Therefore, we can argue that if we really want robust knowledge of the systems that we're studying, we really need diverse models, which are coming from diverse perspectives of the same system. So it's really hard to get data on what people's perspectives are, right? So you can't really quantify, like, how do you view the world? But we do have data on identity, which, as I argued, shapes perspective. So here's actually data from Canada itself. This is the number of Aboriginal people in green and the number of racialized individuals in these different bars. And you can see that the sort of racial and ethnic composition of undergrad students is pretty different than that of the instructors and the professors that are teaching them. So clearly the representation is pretty off. And this is a problem, not only morally and ethically, but also because it really limits our ability to do science. Without diverse identities in science and race and many other axes, it's really hard to take diverse perspectives of the systems that we're trying to study. And this in turn is really limiting our ability to do good science and to understand the world around us. So with that, I will end and I'll be happy to take questions. I want to thank collaborators I talked about, some members of my lab that I didn't get a chance to talk about, and a number of other folks I've worked with and funding sources. Thanks. Any questions?
Yeah, thanks very much, Allison, for a fascinating talk, and especially the last part, educating us about math biology and about you know, this bias in terms of recruitment of individuals at higher levels when you look at the undergraduate level, what's happening in terms of diversity, etc. So that was quite educational. So any questions from the crowd? Actually, I have a question. Two questions. Can I ask you these two questions before anyone else?
Yeah, go for it.
So the first part is when you looked at you know, these two environments, you have pathogen 1 in one environment, pathogen 2 in the other environment. One would argue that actually it would be interesting to look at some sort of a gradient in the level of the pathogen and look at the spatial component of this in the model. So they develop some sort of a spatial temporal model that includes that sort of variation or that gradient in the level of the pathogen in the environment.
Yeah, absolutely. That's actually, that's what I was doing last fall on my Fulbright project when I was in Montreal. I didn't include it here because it's not published yet, but we were saying that you're migrating not just between two discrete environments, but you're migrating along a continual gradient and looking at how, basically assuming that your rate of accumulating parasites changes as you shift up that gradient. So, and then you can actually look at things like, not just are you migrating or not as a binary, but how far are you migrating up that gradient as a function of like how steep and how much it changes.
Yeah.
So stay tuned. Hopefully we'll wrap up
That's
this year on that. Yeah.
I'll keep my second question for later. If someone else has a question, I want to give it someone else a chance, especially the students.
Suresh has a question in the chat.
Oh, Suresh, you want to ask you a question?
Yeah, it's I typed it because it was a bit noisy, but it's okay now. Yeah, I'm curious about the situation where some individuals may have to sacrifice by migrating. Maybe it's easier for you to read it in order to reduce the density for those who are not migrating, right? So, and specifically, I'm interested in who migrates. So that's the whole idea. And if there is susceptibility variation in the population, then depending on the other parameters, sometimes it may make sense for the medium ones to migrate, and sometimes it may make sense for the high susceptibility individuals to migrate, depending upon the parameters. I was just curious if that's something you've looked into or it's interesting.
Yeah, that's great. So the first, I'll answer the second part first because that's easier. We haven't looked at susceptibility at all. We've looked at, I didn't include here, but we've looked at like tolerance to infection, like how well you can tolerate infection, but not heterogeneity in the population. And then to answer the first one, it's not that was that one where I said you can like split the population and lower transmission. It's not that the migrants are sacrificing, it's that it's actually beneficial for them to be migrating, but it's not beneficial enough for everyone to be migrating, right? So it's a case where in any given year, migrating and staying are equivalent strategies, but you're better off if half the population is migrating and half is staying. So everyone would be worse if everyone stayed and everyone migrated, but it's not as much about sacrificing. I think it's like sort of they're equally beneficial, but you do best when only some individuals are playing each strategy at a time, if that makes sense.
Right, but there could be a situation where you are sacrificing, right? Because
yeah, absolutely, yeah.
So that is what I was trying to get at. If the costs are low and you're essentially migrating to improve population fitness, not individual fitness.
I think we wouldn't find that for the ESS approach because we're looking for the like, if you're an individual and you're trying to be a cheater in this population, like, is it susceptible to cheating? I think if you could try to write down one that was like, you want to maximize population size or something like that, and maybe you would get a different answer. But I think the way that we were looking for our approach, because we were looking for what's best for individuals, I don't think that we would find cases like that.
Thank you. Cool.
Any other question?
I should also say, if you're interested in that, like this last piece, I actually have a perspective piece coming out in theoretical ecology this year, if you want to read more about why I think diversity matters for science and for math bio in particular.
Okay. Okay, so let me ask my second question. So if you look at the cost of infection and cost of migration, is there a scenario in which you could actually maximize migration patterns at a specific parameter regime with the CI and the CM, I believe you called it, where you maximize the frequency of migration in such a scenario?
Or like how often you should migrate?
How often, yes. So it increases actually the migration pattern. So it makes it more frequent to prove your point that actually pathogens could be a driver. And so that would be, in my opinion, the strongest evidence that it's the driver for determining migration patterns. Do you understand what I mean?
No, I'm not sure if also in the model, in the cases where we had the region in black, that was saying that everyone migrates every year. So that's like as much migration as we can have in our model. Is that what you mean, or do you mean something more than that? I can go back to that one.
So the black means once you migrate once every cycle, right?
Yeah, so this is like, oh, in black, this is like your probability of migrating in a given year. And so black here means you're migrating every single year. Blue means you're not quite migrating every year. Yellow means you're rarely migrating, and white means
less probably. I see.
So black is like the maximum migration that you can have.
I'm just thinking in terms of the frequency of migration. I mean, you know, okay, well, one could argue that it could be seasonal, but but okay, all right. I mean, it's not, at least not embedded within the framework of your model, obviously.
That you can migrate more frequently than seasonally.
Yeah.
No. We don't allow for that. I think in reality, like, do I think that parasites are actually the thing that drives migration? No, I think they're an important factor that we haven't studied, but I think unless there was some seasonality to it, you wouldn't necessarily see migration on these seasonal cases. So, I mean, I think probably the closest would be like, if you go back to this picture, there's some reason why you have to be in this orange environment seasonally. Like, it's a really good area to breed seasonally. And then anytime you don't need to breed, you're gonna like book it to the other environment and hang out there. So the sort of movement away from that area is driven by parasites, but the reason why you're spending part of the time here could be for some other reason. Like it's a good breeding site, or it's really good for escaping, I don't know, bad conditions or something like that.
Okay. Okay, thank you. Thank you. Any other question?
There's
Just
another
one more
question in the chat.
here. Jake in the chat.
Have I thought about within population variation and migration and infection costs? I mean, only in the thinking about the different infection intensity classes, that's where we thought about it, where like if you, I can go back to that one, where if you imagine that your population consists of different intensity individuals, that these ones have a lower cost of infection if you have low intensity than the high ones. But we haven't really looked at like, are you thinking about like underlying genetic differences that might mean that you have to pay a higher cost or you have less investment in migration or something like that? We haven't thought about those level, that sort of understanding.
Which I'm not really sure how to interpret it, was birds doing molt migrations, where there's many species of birds where if they fail at mating, they'll migrate part way and do their molt sort of away from the rest of the population. And I've just been sort of, thinking about what would that look like? Is that because they have a higher cost of infection because they're spending all this money on molting, all this energy on molting, or?
Yeah, no, that's a great point. I think you could assume that, like, you could say, okay, either you're in, like, you could build a model that said either you're in like a low body condition because you've expended all this energy, and so you're much more likely to die from migration or infection, or you're in a high level of body condition. So you could basically split the population into two classes and look at like your baseline body. Yeah, no, that would be cool to look at. Again, I think like this is a really simple framework, but yet I feel like there's a lot of different things you can explore with it theoretically. So that would be cool to look at.
Cool, thanks. Great talk.
Thanks.
Okay, maybe with this, we'll thank Allison for a great talk and insightful ideas towards the end of her talk. So thanks for coming, and thanks to everyone for attending.
Thank you so much.
Yeah, thanks for the invitation.
Great.